thank you, Vicki, for that kind introduction. And what a privilege and a pleasure it is to be here with all of you at this 21st gathering of the Moebius Syndrome Foundation. Moebius syndrome, I just learned from Vicki, was described in 1887. There's something cosmic going on here because the National Institutes of Health was founded in 1887. So we are all together here having a 125th birthday, and much excitement here at this moment in terms of the science that makes it possible to get answers to many questions that have been really hard to understand. My goal here is to tell you a little bit from the perspective of somebody whose approach to trying to understand disorders has generally been through the lens of genetics, how that is being now pursued for Moebius syndrome at a moment where we do not yet have what you would call a broad set of answers, but a lot of promise to begin to unravel some of the questions about the causes of this disorder. And one of the things I think we're all learning and probably knew all along is that Moebius syndrome may not just be a single condition, but actually quite a number of conditions, if one looks carefully, that share certain features, particularly when it comes to involvement of the face, but also has the characteristic that different individuals may have details that are quite distinct. And that is an important thing to recognize in terms of research, but even more important, I think, to recognize in terms of the best possible kind of clinical care management. So again, my thanks to all of you for being here, and thanks to Vicki for the invitation. My connection to Moebius syndrome began about 5 years ago when I sat next to Vicki McCarroll at a banquet. It was a banquet of the Genetic Alliance, which is a wonderful umbrella group for many different foundations that have been started to try to draw attention to rare conditions like Moebius syndrome, and there are thousands of these rare conditions. And the Genetic Alliance has served in many ways to provide encouragement and resources so that folks like Vicki and her board members can see what other groups are doing to try to raise attention for a rare condition, to get researchers interested, to deal with many, many issues about health insurance and so on. And sitting next to Vicki that evening as she told me her story about how she started this foundation when nobody seemed to be paying attention to Moebius syndrome, and she thought maybe her son would be the only person she would ever meet with the condition, I was inspired by the way in which this effort has grown and developed over the years, and agreed that this would be an appropriate place for research to perhaps work even harder than it had been. And so I learned then about groups that are working on this condition as a research problem, who are represented here at this meeting. Dr. Mimi Jabs and her group, who I've now talked with on numerous occasions. Dr. Elizabeth Engel and her group, both of these researchers represented here by their teams. And I know many of you are having the chance in the course of this meeting to meet with them, to take advantage of their special expertise about the medical aspects, and if you're willing, to participate in research to try to get additional answers. So it's all mixed up here together in a wonderful way in this meeting. An opportunity for information, for meeting other families who are going through the same experiences, for the chance to deal with professionals who know a lot about this condition from having studied it for many years, and the chance to be part of research studies if that's something that interests you. I thought what I would try to do, again through a genetics lens, was to basically give a bit of information about what it is that we're trying to do in terms of understanding the way in which DNA may hold some of the answers to Moebius syndrome. And I know I'm talking to a diverse group, and in here there are some world-class researchers, and there are probably some people in here who are quite glad to say goodbye to their last science class in 8th grade. So I'm going to try to put this in terms that will hopefully have some appeal to all of those groups. And again, if you're in the expert category, apologies if I insult your intelligence. I don't intend to. So we're talking about genetics, and I always like to look at this picture and say, this is really what we're talking about. I mean, this is sort of a funny picture. Notice the outfits being worn by the parents. and the outfit worn by the child. So you are a mix of your parents, as you can see in this couple walking down the beach. The horizontal and the vertical stripes are supposed to represent in this silly photo the fact that each of us gets half of our DNA from our mother and half from our father, and we end up being some sort of mix of those. But I'm going to get a little more specific about how that works than just this photograph. So DNA is, after all, the hereditary material. It is the information that gets passed from parent to child. It is a remarkable way of storing information. It finds its way onto the cover of Time magazine from time to time because we are in the process of reading out a lot of the letters of this remarkable instruction book. By the way, I've noticed that whenever Time magazine puts DNA on the cover, it always seems to be mixed together with pictures of naked people. And I think that's because editors have figured out that DNA really doesn't sell magazines. And they know what does. So here we have, I guess this is Adam and Eve, but we're solving the mysteries of DNA and selling journals at the same time. So why is Time magazine claiming that we're solving mysteries? What's happening anyway? Why is this a special time? Well, first of all, let's talk about why DNA is so special. If you're looking for a metaphor of what DNA is, I think an instruction book is a pretty good one. Your body is made up of hundreds of trillions of cells. Each one of those cells has within it this instruction book located in the nucleus of the cell. And it's that same instruction that was passed to you by your mom and dad back in the day when you were a single cell, which was true of all of us. You got that instruction book, and every time the cell had to divide, and now you're possessed of hundreds of trillions of these cells, that instruction book had to get copied, carrying with it all of that hereditary information that tracks back to all of your ancestors. So that's what DNA is, and that, I guess, makes it clear this is pretty important stuff. Now, DNA is an instruction book, but it doesn't itself carry out the instructions. You need a lot of machinery to make that happen. DNA is stored in the nucleus in a compact form because if you took the DNA inside one of your cells and stretched it out as a contiguous strand, it would be 6 feet tall, just from one cell. And obviously that wouldn't fit very well unless it was nicely compacted. So DNA is raveled up into chromosomes. And here in this cartoon you can see the chromosomes and the DNA spilling out. And the DNA is this remarkable double helix that Watson and Crick figured out way back in 1953 is the actual structure of this hereditary material. And if we zeroed in on it, you would see that it is this double helix where the rungs of the ladder are actually the part that carries information. Because there are 4 possible letters in the DNA alphabet, only 4. Not as complicated as our 26 letters of the English alphabet, just 4 letters. The letters are actually chemical structures, chemical bases that have names adenine, cytosine, thymine, and guanine, and we just abbreviate them ACTG. And I would bet that if I asked 10 graduate students what ACTG stand for, maybe 5 of them would remember and the other 5 would go, oh, I can't quite remember, look it up. Because we always just talk about A, C, T, and G. And what Watson and Crick figured out is that there's a remarkable way for this information to be stored, and that is the rungs of the ladder. A always pairs with T, and C always pairs with G. And what they also noticed in a throwaway line in their 1953 paper was that if you wanted to copy DNA, it would be pretty easy to see how to do that. You would unzip the double helix, but both strands have all of the information. Because of this matching up of A and T and C and G, you can quickly create 2 new daughter strands if you're simply careful about maintaining that match. So again, DNA, the instruction book for all living organisms, from bacteria to bananas to you all. And I might ask you, if you didn't already know the answer, how many of those letters would it take to specify instructions to build a human being? By the way, you would call that the genome. The genome is all the DNA of an organism. How big is the human genome? How many letters? We're pretty fancy organisms, But it can't be an infinitely large number of letters because it all has to fit inside each cell. So there has to be some constraint. Well, it turns out the answer is about 3 billion. 3 billion A, C's, G's, and T's in the right order. You wouldn't want to alphabetize your genome. That would be bad for you. That is what it takes to create all of the biological potential of a very complicated organism like ourselves. And 3 billion is still a pretty big number. If we decided, because it would be historic, that we're going to read the human genome, and I asked each one of you to read for a little while and then pass it on to the next person, and we read it about the speed of A, C, T, G, G, A, C, T, C, C, A, we would be here 24 hours a day, 7 days a week, for 31 years. And you have all that information inside each cell of your body. And we now know that information. As of 9 years ago, we have that instruction book, which is pretty historic when you think about it. We've crossed the bridge into really new territory, having read this information about ourselves. And about a lot of other species, by the way. We now have that complete genome information on dozens and dozens of other species, and we can draw comparisons between our DNA and theirs that can be pretty interesting. Well, again, DNA is the instruction book, but it doesn't carry out the instructions. DNA actually has to be transcribed into another molecule called RNA, which is a lot like DNA, except it's one strand instead of two, and there's a couple of other small differences. But RNA is the message. So RNA gets this information out of the DNA, and it carries that message off to the protein factory, because it's proteins that are going to do the work of the cell. So RNA is the way in which that information gets conveyed. And if all goes well, protein gets made and does the work of the cell. And you're making lots of proteins right now, using the instructions in your DNA instruction book, carrying them off to the protein factory using RNA as that message, and then making these proteins that allow you to do all the things you're doing. Now, how does a genetic problem come about? Well, it comes about because of a misspelling in DNA. That message was ACTG a minute ago, now it's CCTG. So there's been one change in the letter from an A to a C. That means the RNA also has the same glitch, because RNA is essentially a copy of the DNA. And that means that the message going to the protein factory isn't quite what it should be. And as a result, you end up with a protein that doesn't quite do its job, to a glitch. Now, important thing to know, and we're learning more about this all the time, is that there are no perfect genetic specimens on this planet. Many people that I work with who imagine that they are those perfect genetic specimens are often disheartened to hear this part of the talk. But as we've gotten more and more able to read out instruction books, not just of a sort of reference sequence, but of individuals, we're finding that we all have flaws. Something like 80 to 100 significant flaws, where there's a glitch in a gene that clearly would not be good for it, is our usual burden. We're all walking around with that. And in some instances, that may manifest itself in a way that's apparent, in other instances not. My sense is that there are some of those genetic glitches that in a particular location may contribute to Moebius syndrome. But most of them haven't yet been discovered. I'll go through some discussion here in a minute about what we're trying to do to make sense of that. But a very important point is that even somebody who has a diagnosed rare genetic condition probably has a no greater load of genetic glitches than everybody else. It just happens that their particular glitch is located in a place that results in a diagnosis coming to light. We're all walking around with this general burden of things that aren't quite right in our DNA, and it's part of being human.